Hello Year 10, and we are now looking at the effects of pH and the rate of reaction of amylase. Now, if you recall, amylase is a digestive enzyme, and therefore we need to pull on quite a lot of information for this practical, and it will be testing those higher skills, and it will be trying to stretch those of you aiming for a 7, 8, 9, and it will also be pushing those of you doing the foundation paper to see whether or not you're a 4 or a 5. Okay, so these are our learning outcomes today. As always, there is the Caboodle textbook to support your learning. Please feel free to use that whenever you need to. So, just some recapping information about enzymes. Now, remember that our enzymes are a biological catalyst that control, they don't control anything actually. They take part in chemical reactions, speeding them up without being used up. If you think about it, we talk about them being like a key, and the enzyme is a key, and it goes around unlocking the substrate, the biological molecule, into smaller parts. It breaks them up, and just like your door key or your locker key, it can be used over and over again. So amylase is an example of a carbohydrase enzyme. They can talk about carbohydrase or amylase. It doesn't really matter which one they're talking about, but it breaks down starch into glucose. Now, I know that with my group in particular, and if you haven't done this already, try it at home. If you get like a piece of cracker and just chew it in your mouth over and over and over for about a minute, don't swallow it, you'll realise that the amylase starts to act, and that amylase then you start to taste the sugar, you can taste the glucose. So it goes from a very starchy taste to a very glucosey taste over that minute. And the reason that happens is because of our amylase. Now, this reaction is pH dependent, and we're going to go into that in a little bit more detail now. Just as a recap, obviously amylase is made in the salivary glands, and it's also made by the pancreas. We can test for starch using our indicators. So think back to our food test. One of the tests that we can use, starch is iodine indicator, and it will change from our dark orange browny colour to dark blue black if starch is present. So in this experiment, what we're looking to see is the absence of starch. So you can do this two ways. You can either use iodine indicator to see whether the starch is broken down, or you can use another indicator to see if glucose is made. So what we have here is a water bath over here, and that's held at the temperature that we're investigating. In our water bath, we have amylase solution and starch solution, and a stopwatch. And what we're going to do is take a drop of amylase out at regular time intervals to measure how long it takes for the starch to break down. So when we drop it in, we've got iodine in our dimple tiles. When we drop in the sample from the pipette, is there any starch present or not, or has it all been broken down? So here's our equipment. Now, obviously, we need to think a little bit about risk assessments, and in this example, we're going to look at glassware, and we're also going to look at iodine solution. Also, you're going to have not boiling hot water, but you're going to have hot water. So here's your method. It is a longer method. As I said, this is stretching our sort of 6+ candidates in an attempt to make sure you get the best grade possible. This is a nice detailed method, but again, just like with the other ones, try and reduce that down so that you can make it as simple as possible so you've got a chance of remembering it. So what we're going to do is start off with putting 2 centimetres of amylase into our test tube, then only add 1 centimetre of pH buffer, and you are going to, that pH buffer holds the pH at that level without letting it change. Now, I just realised that I said on the previous one that we were going to change the temperature. In this experiment, we're going to hold the water bath at 30 degrees because we know that the enzyme reaction is relatively fast. However, you can change the temperature up and down. Obviously, if you make it too low, it would take a very long time. If you raise it too much, you risk it denaturing. Okay, so while we're waiting, we're going to add a drop of iodine solution to each of the dimples on our spotting tile. And after 5 minutes, using another syringe, add 2 centimeters of starch to the amylase buffer solution and leave it there to test. Then every 30 seconds, what we're going to do is we are going to remove a drop and add it to our dimple tile. And we're going to keep doing that until the mixture remains orange and we do not see any elements of blue-black. Now this can be open to perception, and it is really important that you stir your solution. Sometimes in science we can use an electric stirrer to ensure that happens. Okay, so here's the link to the practical so you can watch it online. It can be done with lots of other enzymes. The other one that they're quite interested in is pepsin, which we find in the stomach, and they're quite interested in whether or not you understand the same principle but apply it to a different enzyme. So our results. Now what you should see is a pH like this. And the pH you can see here is the optimum is at 8 and not 7. So at low pH and high pH, the iodine keeps staying black because the enzyme has been denatured. So these two extremes of my curve, the enzyme has been denatured. And in just a few minutes at pH 7 and 9, it shows us that the iodine is brown, so all the sugar has been broken down. And so it's quite important when you're plotting this that A, you remember it's a curve, and B, you can remember to not go all the way down to 0 because our pH scale doesn't start at 0. So please don't extrapolate to 0 because that will lose you points. And you need to make individual points. And if you're asked to sketch, you don't need to plot all the individual parts on the graph. So if they say sketch, what they're asking for is just draw out what you think. Don't spend ages measuring out. So think about these couple of points. In terms of evaluation, what did we find out? Well, the first thing is that you need to look at what was the optimum temperature, the optimum pH to digest starch. And the actual optimum pH is around 8 degrees, 8 pH 8. However, what you might find when you're doing a practical is that the pH sometimes falls between 7 and 9. And it might be that the fact you need to do more pHs, use more pH buffers to get a more accurate value for the optimum. What could have been possible sources of error? Measuring the solution, timing 30 seconds exactly, and why would the test tubes contain the amylase and pH buffer left with the water bath for 5 minutes before starting the experiment? And that's to ensure that they already reach the correct temperature for the experiment. And lastly, the water bath was set at 30 degrees C and the thermometer was placed in the water bath. How could the method be changed to improve accuracy? Put the thermometers inside the test tubes. By putting them inside the test tubes, you know exactly the temperature of your solutions, and therefore there isn't any room for that to be an error. It is quite often because the water baths, the electric ones, have a heating element underneath, and sometimes they can get too hot. So it's really key to use a thermometer inside the solution. So the other thing you could use is a digital thermometer. Think about that. So just some useful links. This is how this links to the specification, if any of you are wondering what we're wittering on about. And these are all the skills that you're supposed to master in this practical. Right. Take care. Bye.